So good day everyone, our next topic will be the cell nucleus and the control of gene expression. So for our introduction, the cell nucleus is the control center of the cell that holds the DNA blueprint. In this particular lesson, we will discuss how the nucleus regulates gene expression and the process of using DNA instruction to build proteins. We'll also see how DNA is controlled and how cells decide which genes to use. So here are the learning objectives for this lesson. By the end of this presentation, student will be able to first describe the role of the cell nucleus and its components. Next is to explain the concept of gene expression and its importance in cellular function. Next is to explore the mechanism by which gene expression is regulated in eukaryotes. And lastly is to discuss the significance of gene regulation in development, differentiation, and disease. So let us now proceed to the cell nucleus. So the cell nucleus, again, it is the center control of eukaryotic cells that houses genetic materials, the DNA, and organize it into chromosomes. So we have here the nuclear envelope and the nuclear pores. The nuclear envelope is a double membrane separates the nucleus from cytoplasm, while the nuclear pores it regulates the passage of molecules between the nucleus and cytoplasm. So next we have here the DNA or deoxyribonucleic acid, which is the blueprint of life. It is a hereditary materials and a double-stranded molecule with a sugar phosphate backbone and nitrogenous bases, the ATCG. The sequence of these bases encodes genetic instruction, and there are specific regions of DNA that codes proteins or RNA molecules, which known as the genes. So we have here the gene expression. So it is the process by which the information in a gene is used to produce a protein. It involves two main steps, transcription and translation. So we will now talk first about transcription. So this occurs in the nucleus of the cell. The DNA double helix unwinds and one of the strands serves as a template for the synthesis of complementary RNA molecule called messenger RNA. RNA polymerase is the enzymes that catalyze this process by adding nucleotides to the growing mRNA strand according to the complementary base pairing rule AUGC. So after having the mRNA, the newly synthesized messenger RNA undergoes some modifications before it leaves the nucleus. These modifications include the addition of a cap at the 5 end and a poly A tail at the 3 end, as well as the removal of non-coding regions called introns through a process called splicing. So next we have here the translation. So it takes place in the cytoplasm of the cell, especially on ribosomes. The mRNA binds to a ribosome and the process of translation begins. The transfer of RNA or tRNA molecules carry specific amino acid to the ribosome. It's guided by the mRNA sequence. The ribosome reads the mRNA sequence in groups of 3 nucleotides called codons. Each codons correspond to a specific amino acid or a stop signal. As the ribosome moves along the mRNA, it catalyzes the formation of peptide bonds between the amino acid, resulting in the synthesis of a polypeptide chain which eventually folds into a functional protein. So overall, a gene expression involves the transcription of DNA into mRNA in the nucleus, processing of mRNA, and the translation of mRNA into protein in the cytoplasm. So let us now proceed to the importance of gene control. Not all genes are expressed all the time in every cell. tight control of gene expression is essential for: first is the cell differentiation. Specialized cells express specific genes. Examples are the muscle cells versus the nerve cells. Next is it is also essential in development. Genes are turned on and off during growth and development. Lastly, it is important in response to environmental cues. Cell adjust gene expressions based on their environment. So let us now proceed to the mechanism of gene control. So gene control, also known as gene regulation, refers to the mechanism by which a cell can turn genes on or off, controlling the amount and timing of gene expression. So the gene regulation is crucial for determining cell identity, responding to environmental cues, and maintaining cellular homeostasis. Gene control is regulated at multiple levels. First is the transcriptional control or transcriptional regulation. So it is the primary level of gene control that involves the regulation of messenger RNA synthesis. Transcription factors are proteins that binds to specific DNA sequences near the gene of interest and either promote activators or inhibits or repressors. The initiation of transcription by RNA polymerase. Enhancers and silencers are regulatory DNA sequences that can enhance or repress gene transcription respectively by binding transcription factors. We also have here the post-transcriptional regulations. So after transcription, the mRNA molecule can be subject to various regulatory processes that influence their stability and translation efficiency. This includes mRNA processing such as splicing and polyadenylation, the mRNA localization within the cell, and the action of RNA binding proteins that can stabilize or destabilize mRNA molecules. We also have here the epigenetic regulation. So it is a modification such as the DNA methylation and histone modification. It can influence gene expression without changing the underlying DNA sequence. Methylation of DNA typically leads to gene silencing. While the other one, the histone modifications can either promote or inhibit transcription by altering the chromatin structure and accessibility of the DNA transcriptional machinery. We also have here the post-translational regulation. Once a protein is synthesized, its activity can be further regulated through translational modifications such as phosphorylation, acetylation, ubiquitination, and proteolytic cleavage. These modifications can alter the protein stability, subcellular localization, and interactions with other molecules, thereby modulating its function. Lastly, we have here the feedback regulation. So many biological pathways are regulated by feedback mechanism, where the end product of a pathway acts as a signal to regulate the activity of enzymes or transcription factors involved in its own synthesis. Negative feedbacks loops help maintain homeostasis by dampening fluctuations in gene expression, while positive feedback loops can amplify a signal to produce a rapid or robust response. So let us now proceed to the transcriptional control. So transcriptional control involves the interplay of various molecules. We have here the promoter. It is the DNA sequence where RNA polymerase initiates transcription. We also have here the enhancers, the regulatory regions that increase the rate of transcription by binding activators proteins. Next is the silencers, where regulatory regions that decrease the rate of transcription by binding repressor proteins. And lastly, we have here the transcription factors, which is a protein that binds to specific DNA sequences and regulate transcription initiation. So the RNA polymerase is the enzyme responsible for transcribing DNA into RNA. And there are three main types of RNA polymerase present in eukaryotes. These are the RNA polymerase 1, 2, and 3. So the RNA polymerase 1 transcribes ribosomes RNA genes, while the RNA polymerase 2 transcribe protein coding genes and some non-coding RNA genes. Lastly, the RNA polymerase 3 transcribe the transfer RNA or tRNA and other small non-coding RNA genes. So each RNA polymerase requires specific initiations signals on this DNA to start transcription. Let us now discuss the transcriptional control. So we will compare the transcriptional control to a stage play. So we will assume that transcription is the star of the show, and the promoter region is the stage, and so on and so forth. We will see how these different molecules or various molecules interact to regulate transcription. So first we have here the promoter region. So it serves as a stage where the transcriptional machinery assembles. It's like the spotlight that illuminates the beginning of the play. Transcription factors like the stage managers direct the actions by binding to specific sequences within the promoter region. We also have here the transcription factors. So in a stage play, they are like the directors and producers, where they oversee the entire production. They also determine when and how transcription should occur. Some transcription factors act as activators, signaling the start transcription, while other acts as repressors, preventing or inhibiting transcription. We also have here the RNA polymerase. So in a stage play, the RNA polymerase is the leading actors on a stage. They are responsible for reading the script or DNA and transcribing it into RNA. The RNA polymerase follows the directions given by the transcription factors and navigates the DNA sequence, adding complementary RNA nucleotides to the growing RNA strand. We also have here the enhancers and silencers, and they act as the stage set and props. So enhancers are regulatory DNA sequences that can enhance transcription when bound by activator proteins, while silencers can suppress transcription when bound by repressor proteins. Together, they help create the right environment for the transcriptional machinery to perform effectively. Next, we have here the chromatin remodeling complexes. So in a stage play, we will assign them as the stage crew, which they modify the chromatin structure, making the DNA more accessible or inaccessible to the transcriptional machinery. By loosening or tightening the chromatin, these complexes help regulate transcriptional activity. We also have here the epigenetic modifications. So they are like in a stage play, we will set them as the backdrop and the lighting. So epigenetic modifications such as DNA methylation and histone acetylation acts like backdrop and lighting of the stage. They set the mood and atmosphere, influencing whether transcription factors and RNA polymerase can access the DNA. For example is the DNA methylation often represses transcription by blocking the binding of transcription factors. So overall, in this what we call theatrical analogy, transcription control is a carefully choreographed performance where each player has a specific role to play. When these various molecules play together, they determine when and how genes are expressed, ensuring that the right genes are turned on or off at the right time and in the right cells. So we also have here transcriptional factors. So transcription factors are proteins that regulate transcription by binding to specific DNA sequences. So transcription factors have two main domains. First is DNA binding domains, where it recognizes and binds to specific regulatory sequences on DNA, can either be enhancer or silencer. Next is we have the protein-protein interaction domains, where it interacts with other proteins such as the RNA polymerase to influence transcription initiation. So transcription factors can act as activators or repressors depending on their interactions with other proteins. So in order for us to better understand the works of transcription factors, we will again make an analogy where we will assign transcription factors as the matchmakers. So again, we will dig deeper about the works of the transcription factors. So transcription factors are like skilled matchmakers where they search for a perfect match between a gene's promoter region and the transcriptional machinery. So these factors recognizes specific DNA sequences within promoter region and identifies compatible traits in potential partners. Once a transcription factors identifies its target, it acts as a mediator that brings together the necessary players or necessary molecules for transcriptionals to occur. It also recruits RNA polymerase, the enzyme responsible for transcribing DNA into RNA and other regulatory proteins to promote the region. It also thus creating a transcription initiation complex. So transcription factors assesses the compatibility between the gene and the transcriptional machinery. It ensures that the right genes are transcribed at the right time and in the right cells by selectively binding to specific DNA sequences and regulating gene expression accordingly. So transcription factors fine-tune gene expression in response to external and internal cues. They can act as activators promoting gene expression or as repressors that inhibits the gene expression. Depending on the signals they receive from the cell environment. Lastly, the transcription factors can adapt their activity based on the needs of the cell. They can respond to signals such as hormones, nutrients, and stressors adjusting gene expression to maintain cellular homeostasis and respond to changing conditions. So that's how transcription factors works if we make it as an analogy for matchmakers. So we have here some example of transcription factors. So many different transcription factors exist, and each with a specific target DNA sequence and regulatory functions. So some example of these include the LAC repressor in E. coli. It represses genes for lactose digestion in the absence of lactose. We also have here the MyoD in muscle cells that activates genes for muscle protein synthesis. And last is we have the P53 tumor suppressor protein, which activates genes for cell cycle arrest or apoptosis in response to DNA damage.
Let's proceed to post-transcriptional. Gene expression can be regulated after the mRNA transcript is synthesized in the nucleus. Imagine a play. The DNA is the script and the nucleus is the backstage and the proteins are the actors on the stage. Transcription is like copying the script, but after that copy is made, there is still work to do before the actors perform. This is where the post-transcriptional control comes in. It's all in the fine-tuning that happens to the RNA transcript or the copied script after it's created in the nucleus, but before it gets translated into proteins or the actors performing. Here's a breakdown of some key processes in this behind-the-scenes world. First is the RNA splicing, removing of non-coding introns and joining of coding exons from the pre-mRNA to form mature mRNA. mRNA capping, addition of a guanine cap or 7-methyl-guanylate to the 5' end of RNA, enhancing stability and translation efficiency. RNA splicing, this is like the editing the script. The initial copy has a necessary section called introns. Splicing snips out these introns and joins the important parts or the exons together, creating the final mature mRNA. Example, a gene might code for a protein with different functionalities depending on its tissue. Splicing can include or exclude certain exons, resulting in slightly different protein versions tailored for specific needs. mRNA polydenylation, addition of a poly A tail to the 3' end of mRNA, promoting mRNA stability and translation initiation. mRNA polydenylation, this is like the adding stage direction at the end of the script. Poly A tail is attached to the 3' end of the mRNA, which again increases its stability and aids in the translation process. mRNA transport, regulated movement of the mature mRNA out of the nucleus through nuclear pores. mRNA transport, it is the edited and prep mRNA transcript needs to leave the nucleus where the DNA is stored to reach the ribosome in the cytoplasm, where protein building happens. This transport is tightly controlled through the special channels in the nuclear envelope. mRNA degradation, it is where the controlled breakdown of mRNA molecules after a specific lifespan. mRNA degradation, not all the scripts are meant to be performed forever. The cell regulates how long mRNA lasts by breaking it down after a certain lifespan. This helps control protein production levels. By regulating these processes, the cell can fine-tune gene expression and ensure the right proteins are produced at the right time and in the right amounts. It's complex but crucial step that allows cells to adapt to their environment and perform their specialized function. Translational control, decoding the message into proteins. Translation is the process by which the information encoded in mRNA is used to synthesize a protein. Ribosomes are the protein building factories located in the cytoplasm. Transfer RNA or molecules act as adapters carrying specific amino acids and recognizing complementary codons on mRNA. We've seen how mRNA transcript gets prepared for protein production. Now let's delve into translational control, the stage where the message encoded in mRNA translates into the actual building of proteins. This is like putting the instructions from the script or the mRNA into actions to create actors or the proteins. Ribosomes imagines this as the protein building factories on the shop floor. They are the complex structures made of RNA and proteins and are found in the cytoplasm of the cell. Next is the transfer RNA or the tRNA. These are the workers' bees shuttling between the mRNA and the ribosome. Each tRNA molecule carries specific amino acids or the building block of proteins and has an anticodon sequence that can recognize a complementary codon sequence on the mRNA. The decoding process. First is the initiation. The ribosome attaches to the specific start codon, often the AUG, on the mRNA. This marks the beginning of the protein coding sequence. Next is the elongation. Here's where the tRNA or the adapters comes in. Each tRNA with its specific amino acid finds its matching codon on the mRNA. The amino acid is added to the growing protein chain. This process continues with the ribosome moving along the mRNA one codon at a time, adding amino acid based on the instructions. Next is the translational control. While the basic process is straightforward, the cell can regulate protein synthesis at the translation stage as well. Signaling molecules. Certain molecules can activate or inhibit protein translation by modifying components of the translation machinery like initiation factors. Think of this as adjusting the speed of the production line. mRNA structure. Specific sequences in the mRNA can influence how efficient ribosomes translate the message. For example, the presence of hairpin loops might hinder ribosome movement. Nutrient availability. If a cell is low on a specific amino acid, translation may be stalled until that amino acid becomes available, like waiting for a missing part in the assembly line. The key players in translation. Several key molecules participate in translation. Imagine a factory that builds machines or what we call proteins based on instruction. Here's a breakdown of the key players in this translation factory. Blueprint or mRNA. This is a molecule that carries the instructions for building the machine copied from the DNA. It's like a recipe with coded steps. Factory machines or the ribosomes. This large molecular complex is where the actual building of the machines happen. It reads the instructions from the blueprints and puts the machines together piece by piece. Next is the delivery tracks or that what we call tRNA. These adapter molecules are like tiny trucks that carry specific building materials or what we call amino acids to the factory. Each truck recognizes a particular code on the blueprint or what we call codon and delivers the matching amino acid. Building blocks are the amino acids. These are the individual parts that get assembled into the final machine or the protein. These are the 20 different types of amino acids, each with unique properties. But next is the foreman or the initiation factors. These protein molecules are like the foreman who gets the factory set up and starts the building process according to the blueprint. Next is the assembly line workers, or what we call the elongation factors. These protein helpers keep the production in line moving smoothly. They help the ribosome or the factory machine read the blueprint one step at a time and connect the amino acids in the correct order. Next is the quality checkers or the termination factors. These process molecules act as a quality checkers who signal the end production when the entire machine or the protein. The stages of translation. Translation can be divided into three main stages. First is the initiation. The ribosome assembles as the start codon or the AUG or the mRNA with the help of initiation factors. The first tRNA molecule carrying the amino acids methionine binds to the ribosome. Next is the elongation. The ribosome moves along with the mRNA one codon at a time. A new tRNA molecule carrying the next appropriate amino acids enters a site of ribosome. Peptidyltransferase, a ribosomal enzyme, catalyzes the formation of peptide bond between the amino acids. The deactylated RNA without its amino acid leaves the E sites, and the ribosome along with the newly formed peptide tRNA complex move one codon forward to the mRNA or the translocation. This cycle of tRNA binding, peptide formation, and translocation continues until the entire mRNA is translated. Next is termination. When the ribosome reaches a stop codon, UAA or UAG or the UGA on the mRNA, termination factors bind. Just like regulating the volume of a song, translation can be fine-tuned to meet cellular needs. Here are some ways with regulation occurs. mRNA availability. The number of mRNA molecules and their accessibility for ribosome play a crucial role. If mRNA are scarce or masked by regulatory proteins, translation will be reduced. Presence of regulatory proteins. Some proteins act as molecular switches. They can bind to specific regions on the mRNA, either stimulating translation initiation by recruiting ribosomes or blocking it by preventing ribosome assembly. Phosphorylation of initiation factors. Chemical modification like phosphorylation can influence the activity of initiation factors. Depending on the specific factor and the phosphate group location, translation initiation can be promoted or inhibited. Just like regulating the volume of a song, translation can be fine-tuned to meet cellular needs. Here are some ways with regulation occurs. mRNA availability. The number of mRNA molecules and their accessibility for ribosome play a crucial role. If mRNA are scarce or masked by regulatory proteins, translation will be reduced. Presence of regulatory proteins. Some proteins act as molecular switches. They can bind to specific regions on the mRNA, either stimulating translation initiation by recruiting ribosomes or blocking it by preventing ribosome assembly. Phosphorylation of initiation factors. Chemical modification like phosphorylation can influence the activity of initiation factors. Depending on the specific factor and the phosphate group location, translation initiation can be promoted or inhibited. Post-translational control, putting the finishing touches on proteins. After proteins are synthesized, they can undergo various modifications that can affect their function, stability, and localization within the cell. Examples of post-translational modification include phosphorylation, attachment of a phosphate group by kinases, often affecting protein activity, glycosylation, addition of sugar molecules influencing protein folding, stability, and targeting. Next is the acetylation. Attachment of acetyl group, sometimes regulating protein activity or stability. Ubiquitination. Addition of ubiquitin molecules targeting proteins for degradation of proteasomes. The life of a protein doesn't end after translation. Post-translational modification act like the finishing touches on the sculpture, shaping the protein's fate. This modification can be diverse. Phosphorylation, the addition of phosphate group by enzymes called kinases, is a common modification that can activate or deactivate a protein. Imagine a protein with an off switch. Phosphorylation can flip the switch to on, allowing it to perform its function. Conversely, removing the phosphate group or the dephosphorylation can turn the switch off. Glycosylation is the attachment of sugar molecules to proteins. This can affect the protein folding, stability, and the targeting to a specific location within the cell or even the outside of the cell. For instance, glycosylation of antibodies can enhance their ability to recognize and bind to foreign invaders. Acceleration involves attaching an acetyl group to a protein. This modification can sometimes regulate protein activity or stability, affecting their cellular processes. Ubiquination is a aging process where a small protein called ubiquitin is attached to another protein. Ubiquination plays a crucial role in regulating protein turnover and ensuring the proper cellular function. The impact of gene expression. The significance of gene expression control extends far beyond the cellular level. It orchestrates the development of a complex organisms from a single fertilized egg. Genes are turned on and off in a precise timed manner, leading to the formation of a distinct tissues and organs with a specialized function. Within organisms, controlled gene expressions allow cells to become specialists. Muscle cells, for instance, express genes encoding contractile proteins that enable contraction, while nerve cells express genes for neurotransmitters that facilitate communication between cells. Tight control of gene expression is also crucial for maintaining homeostasis, the stable internal environment that cells and organisms require to function properly. For example, cells can adjust gene expression to regulate body temperature or maintain blood sugar levels within a normal range. Finally, gene expression allows cells to respond to various stimuli. When exposed to hormones, nutrients, or environmental signals, cells can activate or repress specific genes as needed to adapt and survive. The impact of gene expression. The significance of gene expression control extends far beyond the cellular level. It orchestrates the development of a complex organisms from a single fertilized egg. Genes are turned on and off in a precise timed manner, leading to the formation of a distinct tissues and organs with a specialized function. Within organisms, controlled gene expressions allow cells to become specialists. Muscle cells, for instance, express genes encoding contractile proteins that enable contraction, while nerve cells express genes for neurotransmitters that facilitate communication between cells. Tight control of gene expression is also crucial for maintaining homeostasis, the stable internal environment that cells and organisms require to function properly. For example, cells can adjust gene expression to regulate body temperature or maintain blood sugar levels within a normal range. Finally, gene expression allows cells to respond to various stimuli. When exposed to hormones, nutrients, or environmental signals, cells can activate or repress specific genes as needed to adapt and survive. Disruptions in gene expression can lead to various diseases, including cancer. Cancer is characterized by uncontrolled cell growth and divisions. Mutation in genes that regulate cell progression, apoptosis or deprogrammed cell death, and the DNA repair can contribute to cancer development. Cancer cells often exhibit altered gene expression patterns, leading to the production of proteins that promote cell proliferation and survival. Just like a malfunctioning orchestra can produce discordant music, disruptions in the Disruption in the tightly coordinated process of gene expression can lead to disease. One of the most devastating consequences of misregulated gene expression is cancer. Cancer is a complex disease characterized by uncontrolled cell growth and division. Several types of genetic alteration can contribute to cancer development. Mutations in genes that normally control cell cycle progression, that the orderly sequence of events leading to cell division, can cause cells to divide uncontrollably. Similarly, mutations in genes responsible for apoptosis or programmed cell death can prevent cells from undergoing self-destructions when they are damaged or no longer needed. Disruptions in genes involved in DNA repair can also contribute to cancer, as they allow mutation to accumulate and potentially lead to uncontrolled cell growth. Cancer cells often exhibit altered gene expression patterns compared to healthy cells. These changes can result in the production of proteins that promote cell proliferation, migration, and resistance to cell death. Understanding how gene expression is deregulated in cancer is crucial for developing new therapeutic strategies.